Welcome friends. In this video we are going to talk about cAMP signaling pathway. We will address questions like what is cAMP, how it is produced, and what are the effects of cAMP on cellular targets. But before we start, I request you please watch the previous parts of this lecture series, because if you do not watch, it would be very difficult for you to understand what we are talking about. Now let's start. In cAMP signaling or any other signaling pathway, the most important protein or molecule around which the whole pathway is based is the effector molecule. What is that? The effector molecule. This is the protein, which is actually giving the results, or we can say it is directly or indirectly related in giving results. In this pathway, cAMP signaling, that protein is adenyl cyclase. So adenyl cyclase is an enzyme which is acting here as effector. Now the question is, what does this enzyme do? In our cells we have ATP, right? This is the energy currency of the cell. We call it as adenosine triphosphate. This enzyme acts on ATP, and convert the ATP to cyclic form of adenosine monophosphate, which is known as cAMP. Now, before we talk about cAMP and its effects further, we divert our attention to something else. The enzyme adenyl cyclase is subjected to both activation as well as inhibition. This enzyme is target to two different types of hormones which produce different results or diverse effects. We explain it in simple words. We have two different types of hormones which are acting on the same enzyme, here adenyl cyclase. Their action on the same enzyme is producing different results. In this diagram we have two pathways which are opposite in nature. On one side we have stimulatory hormones like epinephrine, glucagon or ACTH. These stimulatory hormones bind to the receptors which are specifically designed for them, and those receptors are receptor for stimulatory hormones. The binding of these hormones to their receptors is activating the adenyl cyclase. This side we have inhibitory hormones like PGE1, which is the prostaglandin E1, and the adenosine. So these are inhibitory hormones which bind to the receptors which are particularly available for these inhibitory hormones, and that receptor is receptor for inhibitory hormones. So the binding of these hormones to the receptors is deactivating this enzyme or inhibiting the enzyme. In both cases, the binding of the stimulatory or the inhibitory hormones to their respective receptors is making conformational changes in these receptors, which triggers the release of the G-alpha subunits in both trimeric G proteins, which are attached to this receptor as well as the receptor for inhibition. Now, how these G-alpha subunits are removed from the complex of the receptor and the beta-gamma subunit of trimeric G protein, this is well explained in the previous part of this lecture series. Now here comes the main point, that is, this G-alpha subunit is the one which is actually going to influence the effector protein, and here we have that effector molecule is adenyl cyclase. So we have here two types of G-alpha subunits. In the activation side, this G-alpha subunit is known as G-alpha S. This S is for stimulatory. So this G-alpha subunit, after it is detached from the trimeric G protein and the receptor, will go and bind to the adenyl cyclase, and this enzyme would be activated. And we know what activated adenyl cyclase is going to do. It would convert the ATP to cAMP. On the other hand, the second type of G-alpha subunit is G-alpha I. I means for inhibitory. Okay. So this inhibitory G-alpha subunit also going to bind to the same molecule, adenyl cyclase, but instead of activating it, this would inhibit the enzyme, and the result is no production of cAMP. We are not going to get any cAMP. Now the cAMP, which is produced by the action of adenyl cyclase, transduces a wide variety of physiological signals which produce a lot of effects. A lot of effects means more than one effect. Now the effects which are produced by the cAMP, they are actually mediated by the activation of pKa. What does this mean? The effects which we are going to see by cAMP, they are actually not executed by cAMP. Instead, this cAMP activates the enzyme pKa, that is a protein kinase A, which in turn goes and binds to the target proteins or the enzymes and then phosphorylates them. And then those enzymes are stimulated, then we get the effects. Now those effects or the results, they are of two types. The effects by pKa, they are of two types. The short-term effects and the long-term effects. As the name suggests, short-term effects means the responses which are going to last for milliseconds to seconds, and they would be for longer period of time. In this category, we have many responses, but I have listed out only two here: the glycogen metabolism and the fat metabolism. I have already made videos on these topics in detail. You can check the description box if you want to study about this. The second type of effect that is produced by the pKa is the long-term effects. After the activation of the enzyme goes into the nucleus, wherever DNA is present. In the nucleus, this enzyme activates certain transcriptional factors. After they are turned on, these transcriptional factors initiate the transcription of genes, and hence finally we get our proteins. So this was about how the adenyl enzyme is activated and inhibited, and how we get cAMP from ATP by the action of this enzyme. In the next video we will talk about the action of the cAMP on pKa enzyme in detail. If you liked my efforts, give me thumbs up. Thank you.